In one of my previous videos, I talked about the coronavirus's spike proteins as potential drug targets. Towards the end of that video, I showed some ribbon diagrams, and in this video, I want to talk about those, why they're important, and what role they play in the identification of potential drug targets. Welcome back to my channel. My name is Dr. Anwar Youssef Dunbar, and this is Big Discussions 76, Science and Technology. First of all, please like this video, please share it, and please subscribe to my channel. So in one of my previous videos, I talked about the coronavirus's spike proteins, and I ended that video with a brief discussion on why knowledge of those proteins and similar proteins is important, particularly in the context of identifying drug targets. In that video, I threw up some ribbon diagrams, also known as Richardson diagrams. And as you know, one video spawns several others. And so I thought that it would make for a fun discussion to talk about why those diagrams are important and how protein structures and in some instances functions are predicted. So that's what we're going to talk about here. So as a bit of a warning, this video is going to involve some biochemistry and some organic chemistry as well. But it's going to be at a very high level, and if I've done this correctly, it'll be presented in a way that the layperson can understand it and potentially pass this information on to young science hopefuls, and for individuals in the general public who are just curious about how some of these things work. Much of the coverage of the coronavirus is covering the public health aspects in terms of flattening the curve and minimizing its spread, and in some instances the deaths resulting from exposure to the virus. But there are also significant efforts taking place behind the scenes on the research front to try to understand the virus more and come up with treatments to protect human health. So with that, let's jump in. And before I go on, I once again want to once again acknowledge my brother Amal Dunbar, and just that as I make these science videos in particular, I'm very much leveraging his knowledge and skill set in the area of media and video production. He's taught me a ton about media and video production, and that knowledge set has been very, very critical, particularly in the generation of these science videos, which are which can be very, very complex in nature in terms of content and presentation. So let's start with my trusty DNA strand here. So the structure of DNA is well known, and it's been discussed quite a bit, and it's actually the knowledge of DNA is very useful in terms of predicting protein structures and protein function, and I talked about that in the video discussing whether or not the coronavirus was man-made or whether or not it was a freak of nature. How do we predict the structure and, in some instances, the function of proteins that this DNA actually gives rise to. So with that, I have to take us into a discussion of amino acids, peptides, polypeptides, and proteins. So as you can see, I've switched out my DNA model for my trusty animal cell model. And taking us back to Cell Biology 101, we know that the DNA is housed inside the nucleus of our cells. And what happens is that DNA, through transcription and translation, gives rise to RNA, and that RNA gives rise to the numerous proteins in our cells and in our bodies. So now I'm going to go back and I'm going to come back to proteins and what they do and why they're important. But now I'm going to go back and talk about what the proteins are made of. So our proteins are made of little pieces called amino acids. There are actually 21 amino acids, and those amino acids are to make proteins. They're strung together into larger units called peptides, and peptides are chains of amino acids 50 or less, and polypeptides are strands of amino acids that are 50 or more. And the proteins are actually structures of many polypeptides. So we're talking upwards of thousands of amino acids and larger polypeptide chains. And proteins can have whole subunits and moieties. And proteins can get very complex. And so in terms of how amino acids link together to form peptides and polypeptides and eventually proteins, what happens is, if you recall, each amino acid has an amine group on one side and a carboxyl group on the other side. So what happens is the amine group from one amino acid literally joins together with the carboxyl group from another amino acid to form something called a peptide bond or a peptide linkage. And so when an entire peptide or polypeptide chain is created, amino acids are being linked together this way, so that a chain gets elongated and elongated and elongated, and that's how your chain is formed. So in terms of the front and the end of the chain, depending upon where the chain was started and in which direction everything is pointed, your very first amino acid on that chain is going to have its amine group unbonded to anything, and that's going to be called your or the N-terminus. And then on the back of the chain, you're going to have another amino acid whose carboxyl group is unbonded to anything, and that is called the C-terminus. And that's how amino acids bond together to form peptide and polypeptide chains and eventually proteins. Proteins. What are proteins? So proteins are basically, I think of them as the workhorses or the worker bees in our cells. So proteins can be enzymes catalyzing reactions. Enzymes can be proteases cleaving and chopping up other proteins within the cell. Proteins can be receptors along the outside of the cell, or actually on the inside of the cell in the cytosol. Proteins can be receptors on the nucleus. Proteins can be channels allowing the movement of ions into the cell, and proteins can also be structural molecules like cytoskeletons. So our cells actually have something called a cytoskeleton, which helps provide the structure of the cell. So proteins are everywhere within us, and they do multiple things. So, and there are whole papers, written on this. There are whole chapters written on this, and there are whole classes about amino acids, proteins, and what they do, protein structure and function, enzymology. And their whole research labs that spend time just researching these things. We had to memorize all 21 amino acids, and I think I can remember having to memorize these in Dr. Haywood's class. I don't think she'll mind if I say her name, and that's actually not her full name, but she stayed on us, and she actually tested us on the 21 amino acids. And I also thought about something Dr. Haywood taught us, which was that as scientists, it's impossible to memorize everything, and in some instances, you remember some things verbatim, cold turkey. Cold turkey comes from another professor named Dr. Thompson in our department at Johnson C. Smith University. But some things you remember cold turkey, some things you have to go back and research and look back. Sometimes you have to go back and look back at your notes. So it's impossible to memorize everything. And science is always evolving and changing, so you have to constantly read up on the new developments and advancements. So Dr. Haywood had us memorize all 21. And what's important to keep in mind is that they all have different properties. The amino acids, they're grouped. So you have, so in terms of hydrophilicity, you have some molecules that that interact easily with water because they're polar and they're charged, and you have some molecules that are more hydrophobic, meaning that they don't interact with water very easily. So think about oil and water, vegetable oil. If you pour vegetable oil in water and you shake it up and you mix it, eventually all of the vegetable oil will glob together and it will separate from the water. That's because they have two different properties. Water molecules are very polar, and they bind together using hydrogen bonds. And vegetable oil, those are all organic hydrocarbons. So they don't mix, and they don't interact with water molecules. So amino acids are the same way. Some are charged, so that means some will readily interact with water with a polar environment, and some of the molecules, some of the amino acids are hydrophobic, and so they will seek to not interact with water. And so what we know is that based upon those properties, scientists can look and predict what protein structures will form once they figure out the sequence of amino acids in a protein. So they can predict what the structure of the protein will be based upon which amino acids are present and how those amino acids are strung together in sequence. So something else I remember learning from Dr. Haywood back at Johnson C. Smith University is that parts of the protein, once those amino acids are strung together and they orient the way they're supposed to orient, they can either orient to form alpha helices. So, like our DNA strand, they can form helices where they kind of spiral together, or they spiral, or they can form something called beta sheets, where they form these sheets. I remember Dr. Haywood called them beta pleated sheets. So you want to think about pleats. So polypeptides can either form alpha helices or beta pleated sheets. And that's what ribbon diagrams predict. If you look at ribbon diagrams, for example, I think I presented in that video a ribbon diagram of the coronavirus's spike protein, and also of the angiotensin converting enzyme 2 receptor. you could see that those diagrams were both consisted of, if you look at those diagrams, each one consisted of alpha helices and beta pleated sheets. So the spirals, those were the alpha helices, and the longer, straighter shapes with, and somewhere in those there were arrows. Those were the beta sheets. So you can see that those proteins had multiple structures within them, and then you had loop structures, singular loop structures joining those bigger helices and beta pleated sheets together. So again, those different parts of the protein are called subunits and moieties. So this discussion from this point can go in multiple directions, but for the sake of keeping things simple, I just want to say that based upon the properties of these amino acids, scientists can predict the structure of these proteins, particularly from DNA, because we know, what amino acids are going to, based upon the DNA sequence, we know which amino acids those sequences are going to give rise to. So scientists can predict the structures of proteins and what proteins will be made from a given DNA sequence, and that was actually discussed in the article regarding whether or not the coronavirus was man-made or whether or not it was a freak of nature. So how is this information used? So once again, once you predict a protein's structure, and a protein can be an enzyme, and it can be a receptor, or it can be a channel. Once you understand those structures, you can design molecules to, for example, block an enzyme's function, or in some instances, you can design structures that will enhance an enzyme's function. You can blockade a receptor so nothing can bind to it, and in some instances, you can interact. You can create a molecule that will interact with the receptor that will make it more active. The same thing with a channel. You can create a molecule that will, if you know the subunits and what binds to those subunits of a receptor, you can create molecules that will bind to those sites in such a way that will force that channel to stay open, or you can create molecules that will bind that receptor that will make that channel close. And these things all give rise to the effects on the organ level and the effects on the whole organism level in terms of pharmacology and toxicology. So that's what I wanted to say about protein structures and function and ribbon diagrams, and why that's important, and what this information is used for, and how some of this information is being used right now in this whole coronavirus / COVID-19 pandemic / crisis. So let me know what you think in the comments section below. Let me know if you have any questions or comments or suggestions. And again, whenever I make these videos, one video usually spawns another or several. Please like this video, please share it, and please subscribe to my channel. And always remember that your attitude determines your altitude. Take care, and I'll talk to you the next time. Bye-bye.